Before we get into today's material, I want to talk a little bit about an assignment that you had that might have given you some problems. Some of that's my fault, but some of it's your fault. So I want to go over that assignment, which is part 3 of the autonomic system. Now, the part of it that's my fault comes from something that didn't quite register for me at the beginning of this summer. We just recently switched to a new guy that does the Blackboard on campus. So I'd been working with this fellow for some other guy for a long time, and had gotten sort of that's what I'm looking for. Into a routine. And so switching to the new guy, I didn't think about how some things would have to be different. This new guy, he's been actually on campus for a while in a different capacity. So he's familiar with things. He just was never part of the discussions I had with the old Blackboard guy to how I like to do things. And so what he did is he took the summer courses from last year and just copied them over for me to use this year, which is a sensible thing to do in general for how things are run on campus. But what I do is, as problems come up, I change the courses. So if there's a question that needs to be modified, I change that. I don't go back and change it through earlier iterations of the course. I just do it in whatever the current course is, and then I'd have the Blackboard guy copy that course that has all the changes in it for the next semester. And that didn't happen this time. So this assignment actually got changed over the course of this past semester to deal with one of the questions not being perfect. And that change didn't get translated over to the new course because this is the version that I had last summer. But that's not necessarily all of the problems that people had. So I want to talk about this assignment a little bit because it'll also give us a chance for you to understand how to, look at some of the questions that I ask and how to make sense of what I'm looking for. For the ones that I've looked over, I think this first one, and I hope you've realized, because you've probably done this particular assignment a bunch of times, that each time you take an assignment, it sorts the question. So what's the first question one attempt isn't necessarily going to be the first question each successive attempt. So, oh, actually, before I get into this, let me just make sure I got. January, Danielle. Danielle? Amy. Okay. Nikki. Kimberly. Nellie. That's what I thought. Okay. Lynn. Yeah. Jalissa. Yeah. All right. So if I didn't call your name, I know you are. So it's okay. I think most people got this first one. It's just a matter of paying attention to which neurotransmitter is controlling which different things. The next one tends to give people a lot of problems, and it largely comes from the fact that they don't quite get what the sentence is doing. And there are some clues in it as to what's going on. So what the sentence is asking you to do is to describe the components of what controls the constriction of the pupil. Okay. Now for just a second. Okay, so for the autonomic system, everything's a reflex, so there's a sensory component and a motor component to it. But really for the autonomic system, we pay attention to just the motor component. Okay. Controlling the pupil, there's two reflexes. There's the one that constricts the pupil and one that dilates the pupil. And they both have the same sensory input. It's just the amount of light getting to the retina. And that's not terribly important. So when we're talking about the autonomic system anatomy, we're really just paying attention to the output, the motor side of things. So when we're talking about what's constricting the pupil, we're looking at what comes out of the central nervous system to cause that to happen. So the first thing you need to do when you're looking at something like this for the autonomic system is figure out which division you're talking about. So which division, sympathetic or parasympathetic, controls constriction of the pupil? Parasympathetic, which is not one of the things here. But knowing that, then you can work towards finishing this. So it's asking, where is the neuron that sends the signal to the ganglion? That means the preganglionic neuron, or the preganglionic fiber. Some people put ciliary ganglion in there. Okay, why is that not correct? First two lines. Sorry? No.
It's sending it to the ganglion.
Right. It's saying it's going to the ganglion. It's the preganglionic neuron. Preganglionic neurons aren't in ganglia. The ganglion that's mentioned in that sentence is actually the ciliary ganglion. So that's just the clue to what I'm looking for there is it's sending to the ganglion. So what's before the ganglion? Now, where are the neurons of the parasympathetic system, preganglionic neurons of the sympathetic system found in general? No, physically, where are they found? Anatomical location.
Axon.
When I'm asking where a neuron is found, I'm asking for a structure within the nervous system. Nucleus is a reasonable thing to say there because we're talking about central nervous system. That's where you define neurons in the central nervous system, but that's far too general a term. Because anything in the central nervous system would be considered to be found in the nucleus. Where are the neurons of the parasympathetic system found? And while nucleus is a reasonable way to go thinking about that answer, it isn't actually about the nucleus, but just the region of the nervous system where it's found. Think about when I was standing over next to that model there, and I was saying where the parasympathetic system is. Brainstem, and for the completeness of the question I'm asking, what else? Not just the spinal cord. Not thoracic, parasympathetic side. Sacral. Which of those two is likely to contribute to controlling the eyeball? Brainstem, yeah. Sacral spinal cord is really for the pelvic region. So, in fact, those first two blanks can just be completed with brain stem, those two words. If you did go with nucleus, the second blank could be nucleus, and you could put brainstem as one word in the first blank. It's more appropriate to say brainstem as two words, but I put both in there. Over the past year, people started to answer that blank, at least the first blank, a little bit more specifically, and to say exactly which nucleus that was, which is technically in the textbook, except that it's misspelled in the textbook. Does anybody know what the nucleus that's mentioned in the textbook that controls pupillary constriction is? Anybody get that?
Yeah.
And how did you spell that?
EDPINGER-WSTPHNL.
Yeah, there's only one D in that.
Oh, that's great.
But regardless, like I said, that's something that people started doing in the past year. So the question we're looking at here isn't coded to accept that anyways. So, yeah. But the neurons located in the brainstem or a nucleus in the brainstem, I think it might be coded to have accepted oculomotor nucleus, which the Edinger-Westphal nucleus is part of. But whatever, that's where that's located. And then it tells you that it goes to the ganglion. And it's not asking for the name of the ganglion, but it's asking where that ganglion is. It's close to what? Now, this is parasympathetic system, so all the ganglia are close to what? No, that would be sympathetic. To the target organ. So what's the target organ that we're talking about here? Yeah. So sends a signal to a ganglion close to the eye. And I think I put in there orbit, which is actually where the ganglion's located. I might have put iris in there, which is a more specific way of saying the target organ, something like that. And then this last blank is often what gets people the most, okay? Which then directly causes a blank muscle to contract. What did people tend to put in for that?
Smooth muscle.
Everybody put
That was
smooth?
the only one I got right. Just wanted to let you know.
And why would, okay. I thought I looked through that and seen some other answers put in there, but I guess you're all just, you figured out what the correct answer is there. What's the clue in the sentence there that points to if you should be putting the word smooth in there?
Is this a particular muscle?
And why do you know that it's asking for a type of muscle?
This is like a muscle.
Because it says a blank muscle.
Yeah.
If it had said the blank muscle, then you would probably put in the iris muscle or this. Shoot, what's it called? No, not radial. Radial is the one that dilates the pupil. I can't remember what the name of the other one is. But anyways, yeah, the fact that it has a before the blank is asking for something kind of general. Okay, so it's asking for a type of muscle. Okay. And the reason it's in there is that when we're talking about the autonomic system, it controls smooth or cardiac muscle. And obviously it's not cardiac muscle here, but so that's the idea. So I wanted to go through that in some detail so that you can kind of unpack how the sentence is put together and the kind of answers that are looked for. I'm not going to go through the next one in quite as much detail, but I do want to be sure that people understand part of what's going on here. So it's asking about fibers that are branched, preganglionic fibers are branched, blank, blah, 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 blah. While the preganglionic fibers of the blank division tend to innervate just one target. That should say one, not on. I never caught that typo. So the first and last blank in here are really just asking for the two divisions of the autonomic system. Okay. So I did look at somebody's answer when they texted or emailed me for help, and they put the same division in both blanks. Okay. So parasympathetic or sympathetic, whichever it was, put the exact same thing in both blanks. And just the way that it's worded, it's like preganglionic fibers this, while preganglionic fibers that. So it's got to be two different things. A lot of these multiple blank sentences are going to be set up that way. There's going to be two things. Okay. In the special senses one, there was one that was asking about special senses and general senses. Okay, so the two blanks there were sort of a pair that went together. And you'll see that kind of thing a lot. So pay attention to when the sentence is set up to be a little bit reflexive like that, to have, in this case, the two opposing divisions mentioned there. In between, the other three blanks are separated by hyphens. So it's a three blank phrase that's hyphenated. And it's talking about the response of the division that the first blank is about. Okay. So sometimes people get a little hung up on that, but these little lines here between the blanks are hyphens. And you just recognize that. Now this next one is the one that probably got the most accurate cursing of my name.
No, we didn't curse you, we just thought you were trying to kill us. Yeah.
Well, if you don't curse people that are trying to kill you, then you don't care about your own life. Anyways, so this is the main question that's the issue about this using last summer's course again. For this question, there's three things here. Two of them are in the book. Okay? The third one's not in the book. What I used to do, and I just got away from it because of how I present the material eventually, I used to cover the third one in class, which actually I kind of like setting up the question that way. Because sorry?
You like to make us cry.
Well, if I'd done what I used to do in class, then you shouldn't have cried so much. But it would be a question to make sure that you're getting material out of the reading material and the class material. But I didn't set it up that way. In discussing the case, the third one would have come to light. Now, what this is about is that there are exceptions to the general idea of how the autonomic system is set up neurotransmitter-wise. All preganglionic fibers release acetylcholine onto the ganglia. The ganglia have nicotinic receptors, which is described in the book, and they're always activated by the preganglionic fibers. And then the postganglionic fibers release different chemicals onto the target organs because one target organ is going to get two signals and it has to be able to tell them apart. The heart, for instance, when it gets a norepinephrine input, is going to speed up. And when it gets an acetylcholine input, it's going to slow down. That's all the heart knows. It just knows what chemical is being released because of what receptor is being bound to. But there are some exceptions that basically come from the fact that not everything is a straightforward two-input opposing process. And for the first two, it's really about the ones that don't have parasympathetic input. Okay. So what does not have parasympathetic input? Or what are the first two things in the list there? The ones that are out of the book. Yeah. So sweat glands. They don't have any parasympathetic input. They don't need to worry about what the chemical signal is. Any chemical signal arriving and activating their receptors is going to have an effect. Okay. And so the most reliable sources I've been able to find say that that acetylcholine is released by post-ganglionic sympathetic fibers there, which is an exception. Okay. What's the other situation where there's not a parasympathetic input? Yeah.
Blood vessels.
So blood vessels in general. Okay. So when we're talking about blood vessels, now the chemical signal is important because for a blood vessel that really only gets sympathetic input, the sympathetic system, which has to tell all blood vessels do something at the same time, just has to tell them different things. If it releases norepinephrine, it's going to cause the blood vessel to constrict. Okay. If it releases acetylcholine, it's going to tell the blood vessel to dilate. Okay. So which blood vessels have to dilate during the fight or flight response? Yeah. So when you're trying to run away from the lion on the savannah, you need more oxygen going to your skeletal muscles. So blood vessels in skeletal muscles. Yeah. Also get acetylcholine input. Now there are some blood vessels that get parasympathetic input. They're very rare, but they work on the same concept as what's going on in the skeletal muscles. So then that leaves the third thing, which the book doesn't address. And now I wrote the chapter in the book, and so I didn't put this in that chapter. Part of the reason I didn't put it in the chapter is that the first two are better documented, and the third one here I left to my class material just so that it wasn't part of the published book. And in the case study, what I used to have is another part. So this is, the retail manager who got dizzy. There's another part. What actually sent him to go see the doctor was he wanted to ask about Viagra. And the first question that the doctor asked him was, well, do you have a problem getting an erection? And he says, no, that's not the problem. I have a problem ejaculating. And so he said, well, that's not what Viagra is about. So from that, talking about it in the case, it would come out that in the reproductive system, the autonomic system does not have that opponent input to things. Instead, the sympathetic system, or I should say the parasympathetic system causes the erection, and the sympathetic system causes ejaculation. Those are not necessarily required to occur together, but for optimal functioning of the external genitalia, they do go together. So that third pair of blanks would be external genitalia. Now, I took that Viagra part out of the case, partly because the other symptoms in the case are mostly upper thoracic spinal cord functions of this sympathetic system, and controlling reproductive function would be upper lumbar region. And so it'd be anatomically, it was just the wrong area for a symptom to show up. So remember, the guy was having neurons dying off because of alpha-synuclein toxicity. And all of the symptoms that I had in the case would really be localized to this part of the spinal cord up here. And if it were a reproductive function, that would be down here. So it wasn't a proper case to have a little bit of, most of the symptoms localized to the upper thoracic region and one to the upper lumbar region. So I just took that part out. And in taking that part out, it also kind of messed up this question. And since it went back and got the question from last summer that I didn't update. So that's why it was in there. So for this one, if you want to go back and optimize your score, just type in external genitalia, and that'll give you those points there. Now, the last question here is another one that gives people problems. And this is another one where you have to really kind of pay attention to how the sentence is set up and what you're trying to get out of it. So it starts off talking about smooth muscles and digestive system. And that's what the sentence is about. Everything in the sentence is about smooth muscles and the digestive system. So for the most part, they're stimulated to contract by one of the divisions. Obviously parasympathetic, because it says sympathetic later in sentence, except for blank, which are stimulated by the sympathetic division. So what this is asking you is what, when your digestive system is active, the parasympathetic side is causing muscles to contract and digestion to take place, pushing food through the system. But there's a special type of muscle in the digestive tract that is controlled by the sympathetic system. When it contracts, it keeps digestion from taking place. That's what it's getting at.
So you're actually looking for a thing with digestive muscle.
Yes. Somebody had put in blood vessels or something like that, which I can kind of see the logic behind that answer, which is when your sympathetic system is active, then blood vessels will constrict, reducing blood flow to the digestive system, et cetera. But it's not as straightforward, or not. It's not as, I can't think of the word I want to say here. It doesn't fit in the sentence quite as well. It's not necessarily about the smooth muscles in the digestive system. Also, if you wrote in blood vessels, it'd be 2 blanks, and there's only 1 blank there. Okay. So you're looking for a type of muscle that the sympathetic system controls, and it's going to have the opposite effect of the parasympathetic system. It's going to keep digestion from taking place. Okay. So that's what that's getting at. I think the biggest problem with this is people see this blank and they don't realize it's still talking about smooth muscles in the digestive system. So that's what the sentence is getting at. So, okay, so I just want to go through those in some detail and also point out for the third part of this question's answer, type in external genitalia. I didn't give you that information. Oops. In class, and it's not in the book, and that'll complete that answer. And the other parts are definitely in the book. So you should be able to complete that. Okay. So today we're going to start the cardiovascular system, and we're going to do it this week and next week, which isn't entirely enough time for it anyways, but it's the time we have available. So we're going to start with the blood today and get to the heart, and then we'll finish the heart next week and talk about blood vessels also. I think it was in April, I was reading this book. It was really pretty incredible. There's this sort of historical book about, well, it's focused on the heart, but the cardiovascular system, and sort of how a lot of modern medical techniques have come to be. And so it was talking about the history of heart transplant surgery and that sort of thing. It was discussing cardiovascular disease, and it made a really, did a really good job of explaining atherosclerosis in a lot of detail. And I'd like to include that in talking about blood vessels. I don't know if I'm going to have time to do that this summer, so I might not get to it, but it was really kind of interesting. And it also, I might not talk about it with the actual blood vessel stuff we're doing next week, but it also has to do with immune function. And so the immune system chapter comes after the cardiovascular stuff. So I might use that as sort of a bridge between the two a little bit when I get to that stuff. Anyways, in this folder, you have the part 1, 2, and 3 assignments for all 3 chapters, the blood, heart, and blood vessel chapter, or vasculature chapter. They are not all due by next week. I'm actually, I don't think I have the due date set up the way I'm about to describe them, so I'll get in there tomorrow and update the due dates to reflect what I'm about to say. We're going to do the blood today, so the blood chapter and the blood lab quiz will be due for next week. And then we'll start the heart stuff today, but we won't finish it. And then we'll be looking at the vasculature next week. So heart and vasculature will be due the following week, which I don't have a calendar in front of me, so I don't have the dates, but they'll be due 2 weeks from yesterday. And I kind of doubt it, but I might get to talking about the heart and EKG lab material today. But I'll probably have that due with the other stuff. I actually really kind of doubt that I'll get to the EKG stuff today, but the lab quiz here will also be due 2 weeks from yesterday. So, but I've put everything in one chapter, one folder. It used to actually be that the blood stuff was in the endocrine system folder. What I did last summer was I did the autonomic system separate from the endocrine system. So like last week I had it all together. If I did what I did last summer, then we would have done just autonomic system last week, and we did endocrine system and blood this week. I decided to switch it up so that we had all of the cardiovascular system over these 2 weeks. So we'll go ahead and do that. For the blood, what we're going to concentrate on is doing the blood lab material this morning, which I've got some stuff set up in the back, and so we'll do that. Oh, shoot. And then in the second half, I'll probably have to do a little bit more blood stuff after the break. And then we'll start looking at the heart a little bit. Monday, I went to a really kind of cool workshop. We had we had a chance to buy some physiology equipment a year ago or more, but we were never really good at using it. I tried using it in a lab a little bit. So I actually got to do that Monday, and the exercise that the workshop had us doing, or one of the exercises, was to record an EKG. So we only have one set, so I can't have everybody do an EKG, but I'll use that, and that'll probably be at the beginning of next week to show you something about the EKG. So, anyways, where are we here? What I want to start off with talking about blood is what blood is, what makes it up, and that's what the lab exercise is really going to be about. The chapter starts off with an idea of what blood's functions are. It's the medium through which everything's transported. It has a lot to do with defense. White blood cells obviously have blood in their name, but they're also the main players in the immune response, which we'll talk about in the immune chapter 3 or 4 weeks from now. But being in blood, it kind of implicates blood in that. And then blood is very important for various things, for maintaining homeostasis. There are all sorts of blood whatever levels that we regulate, like blood glucose or blood calcium, those sorts of things. So blood plays a part in that, as well as blood clotting could be considered a homeostatic mechanism, keeping the blood circulating around. So we'll get to talk about that a little bit later on. What's most important for what we're starting off with here, is what the composition of blood is. This is a test tube of blood, represents a test tube of blood, that's been separated out by centrifugation. So imagine this pen here is, you guys good?
A test tube.
I'm embarrassing them for talking to that girl. Thank you. So imagine this is a test tube. It's been filled with however many milliliters of blood that represents, put in a centrifuge and spun around. Okay. The centrifuge spinning around exerts the tube to a high level of force, some numbers of times the force of gravity. And in doing this, it'll separate the blood into its major components. You don't need a centrifuge to do this. If this was a test tube of blood and we just put it on the counter, left it overnight, and came back, we'd see the exact same thing. So the force due to gravity would do the job too. It just needs a long time. To do this in a rapid manner, then we use a centrifuge to do it. The components in this picture are listed as if there are 3 things, but really we think of it overall as 2 things. There's the plasma, which is the extracellular component of blood, and then there's a cellular component of blood, which is often referred to as formed elements. It's kind of an odd term, but it's referring to the fact that blood cells are formed, actually not in the blood, but in bone marrow. And that's going to include white blood cells and platelets, which here are referred to as the buffy coat, and red blood cells, which here is referred to as the hematocrit. So the two bottom things are the cellular component of the blood. There's 3 types of cells that we associate with blood. When we separate things out, the hematocrit, the red blood cells, is the red component because the blood cells are red. And then the buffy coat, which we see between the red blood cells and the plasma, is referred to that because it looks like it has a buff color to it, so to speak. And it's just a coat of cells that's on top of the red blood cells separated still from the plasma. It is mostly composed of white blood cells, so it basically has the color of white blood cells. Which is, you're probably most familiar with as pus. That's basically what it is. Okay. But it just has its own little name there. To see these three things really is a matter of this volume of blood. For a smaller volume of blood, like we'll be working with, you won't actually see the Buffy coat. There's not enough of it there to make out, but they're highlighting it here. Now at the bottom, it's that first tube is labeled normal blood, and it shows that there's two different ranges for the hematocrit. Women can have 37 to 47% of their blood volume that's just red blood cells. Men can have 42 to 52%. It's just that. So whatever number is actually determined to be the red component here, and when you can't see the Buffy coat, it's really just the whole thing. Whatever that is, that's the hematocrit number, and it represents the volume of your blood that is composed of those cells. Okay. And so the rest of the blood, so if we take the hematocrit number and subtract it from 100, 100 representing 100% of your blood, we take out the hematocrit number worth, what's left over is going to be the component that's plasma. Now, usually the overall average number for the hematocrit that we work with, just partly to be nice and round number to work with, and it's fairly close to the actual average across all people, not just separated into gender, is about 45. So if the hematocrit number is 45, what's the plasma composition of blood going to be? Which is?
100 minus 45.
Right, what's 100 minus 45?
50.
55, thank you. So plasma is about 55% of the blood, and the cellular component is about 45% of the blood.
Is that just for adults, or is that the same like with children?
This class is just studying normal adult anatomy and physiology, so I don't know anything about children. Now, the way they have this bracketed out in this picture with the Buffy coat being separate is a little misleading. The Buffy coat is not ever enough of what's in the blood to even register number-wise there. Red blood cells are about 99% of all of the cells in the blood. And so the white blood cells and the platelets, if you can see them, if there's enough of a volume to make out a Buffy coat, are still less than 1% of, well, yeah, less than 1% of the cells in the blood, and a mere fraction of 1% of the overall blood volume. So we don't usually deal with the Buffy coat, or more likely we'll just consider it mixed in with the hematocrit number, especially when we're working with a small volume of blood like we'll be doing today. So the 45-55 split is, again, a very average number, and there's some variation as we see in the numbers at the bottom of that picture there. So cells are cells. We'll talk a little bit more about those as we go through. And the plasma is mostly water, and that's why blood is liquid. And dissolved in that plasma are going to be other components. There's a component that are called plasma proteins, specific proteins that are in there. Their primary reason for being there is to make the plasma thicker than water. Okay, so the old adage that blood is thicker than water, which is supposed to mean family is most important, actually is referring to the chemical composition of plasma. The plasma proteins make it thicker. Not on the same scale, but think of the white part of an uncooked egg, okay? That is a water solution with a high content of protein. It's a higher content of protein than plasma. Plasma is not that thick, but that's the idea that proteins make the watery component of blood thicker than just plain old water. Now, I use egg as an example there actually for a very important reason. The protein that's in egg, called albumin or ovalbumin, is the protein that's in plasma. Now, I said albumin or ovalbumin. Albumin is just this general name for this kind of protein. And eggs have them, fowl have them, and humans have them. Ovalbumin is referring specifically to the version of that protein that's in the chicken genome. So it's found in chicken eggs, that sort of thing. It's not called ovalbumin when it's in humans. It's just called albumin. But it's mostly there just to make the plasma thicker. Okay. But then we're also going to see nutrients like glucose, amino acids, and oxygen, and micronutrients like vitamins and minerals in the blood. Hormones, which we kind of talked about last week, travel through the blood. And then the et cetera covers a whole bunch of other things. Waste products coming from tissues to be cleared out, carbon dioxide and other things that come from the everyday metabolism of cells. Regulatory signals, which aren't exactly hormones, but they can have a role to play there too. There's a bunch of things dissolved in plasma. Depending on the exact source you get, the numbers are going to vary a little bit, but how it's described. I prefer to use the least precise numbers possible. You'll often see sources saying that plasma is 91.5% water, something like that. But that actually can vary quite a bit over the day for a single person. So it's more accurate to just say that water is about 90%, plasma is about 90% water. Okay. 91.5 is just too precise a number considering how much an individual's hydration state can change over the day. Now, you had a little bit of trouble with the subtracting 45 from 100 math that I just presented to you, so this is really going to blow your mind. If plasma is 90% water and blood is 55% plasma, how much of blood is just water? Not a very good guess.
What'd she say?
She said 40.
90% water.
90% of 55 is 90% of 55.
35.
You're just guessing, folks.
What do you want, percentage?
Yeah. So I'm asking you what percentage of blood overall is water. And the way you figure that out is we know that plasma is 55% of blood and water is 90% of plasma. So 90% of that 55% will give us that. So what is 55 times 0.9?
49.5.
Yeah, 49.5. I would have gladly accepted 50. Nice rounding number, but yes, more accurately, this 49.5, which means that blood is half water, okay? Just water, nothing, everything else separate. Okay? Which gets at the point that it's a liquid tissue, okay? Because it's got so much water in it. Now, I said earlier, what we have here, we can spin in a centrifuge and get that, or we can just leave it out on the counter and overnight it'll separate like this too. So it doesn't matter exactly how much force is being applied. As long as the blood's not moving around in the circulatory system, it will separate into these components, okay, just naturally. You've probably seen that effect in a different situation, but it's the exact same idea. If you have, an oil and vinegar based salad dressing, and you put that bottle down on the table, what happens?
It separates.
Separates into what?
Oil.
What does it separate into?
Oil and vinegar.
Okay, what is vinegar?
Mineral?
It's an acid, which means that it's what kind of solution? Water-based solution. So water and oil separate. Which one's on top? Oil. Why? Okay, I'm gonna take a bowling ball and feathers and put them together. Which one's gonna be on top? The heavier one's gonna be on top? Lighter one's gonna be on top. So oil is lighter than water. Okay. It'll float. Okay. Ice floats on liquid water because it's lighter. Now, lighter is not a very accurate term to use in the scientific sense. It's more appropriate to talk about density. Oil is less dense than water. So when we're looking at the components of blood, they separate out naturally because of density. What is the most dense component of blood?
The red blood cells,
Right. Why do you say that?
because they're on the bottom.
Right. Simple as that. They're on the bottom. Which means that red blood cells are heavier, so to speak, than white blood cells or platelets. Now, platelets are actually these little cell fragments. Now, saying that they're cell fragments isn't my way of setting up a trick question. They are cells. They're parts of cells. But they're very small. They're like 1 or 2 microns in diameter, micrometers in diameter. Red blood cells on average are about 8 micrometers in diameter. White blood cells are different types, and they can range in diameter from about 12 micrometers to 20 micrometers. So which is the biggest type of cell? White blood cells, because they can be as big as 20 micrometers in diameter. But they're not on the bottom. Being biggest does not make them heaviest, does not make them most dense. What is it about red blood cells that makes them more dense? Platelets are a different type of cell. What do red blood cells have that makes them more dense? No, they'd have the same water concentration, if you would, as any other cell. What do red blood cells do? What's their job?
They carry nutrients.
Not just nutrients, they specifically carry oxygen. How do they carry oxygen? No, inside. What do they have inside that helps them carry oxygen? I think I heard it. Hemoglobin, right. Hemoglobin's a protein, but it contains something that makes it possible for the oxygen to bind to it. Now, does anybody know why blood is red? What?
It's the amount of oxygen.
Yeah, it's because of oxygen, but it's not, oxygen's colorless gas. What gives blood its red color? Why does oxygen being present make it red? Does anybody take any supplements to help having an anemic condition? Iron. Oxygen binds to iron. That's what's in hemoglobin that makes it possible. What do we call the compound, Not worrying about blood for a second. What do we call it when iron oxidizes? Rust. Yeah, iron oxide. That was a compound, but that's not what it's. But yeah, rust. What color's rust? Reddish. It's exactly the same thing as what's going on in blood. Iron in blood oxidizes, and it gives us this red kind of color.
So that's the only reason why it's red is because the iron in the blood?
Right. In some science fiction universe where blood's different, that instead of iron it uses, I don't know, copper or something like that, then blood would be a different color. And if it's copper, I think that would make it green. So maybe that's what Spock is all about. But so, yeah, it's the color is just the iron-oxygen reaction. Now, when oxygen is not present, blood's still kind of red. It's a darker, more purpley kind of red, which sometimes people think of as blue, which is the color we associate with deoxygenated blood. But so it's not just the oxygen and iron. Iron has inherently reddish color, and it can still kind of have that even when it's deoxygenated. Now, every red blood cell has a bunch of hemoglobin, about 280,000 molecules of red blood of hemoglobin in a red blood cell. Each hemoglobin molecule has 4 iron ions in it. So that means that there's over 1 billion iron ions in any given red blood cell. Iron on the periodic table is much further down than carbon, oxygen, nitrogen, hydrogen, all the things that make up proteins and cells and that sort of thing. So it has a lot of heavy metal in it. Now, 1 billion isn't a really big number for chemicals, but it's still enough to give red blood cells the density to be heavier than bigger cells. So it's going to be at the bottom. Okay, so that's what the hematocrit's about. We're going to do this, and the process of doing this does not really lend itself to